you
decision.
I will tell you.
Bismillahirrahmanirrahim. Good morning, good afternoon, good night, good night according to your time. Distinguished guests, honorable participants, and honorable speakers from around the world. Inspired by Michael Jordan quote, learning is a gift even when pain is your teacher. We remind that growth comes through effort and challenge. This spirit of sustainable effort, learning, and innovation, diverse scientific discovery, and sustainable development, turning challenges into ideas and ideas into lasting solution for better future. It is my great pleasure to welcome you all the comstech workshop on blue biotechnology for health and environment. This workshop aims to explore innovative science-based solution for harness the potential of blue biotechnology and advance human health, protect our environment, and promote sustainability. We are honored to bring together a diverse and dynamic audience of policymakers, researchers, academicians, students, and experts with participants from voice member states and beyond. Without further delay, let me invite to all of you to join me for the official opening of this webinar, beginning with recitation of Holy Quran.
أعوذ بالله من الشيطان
الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Ladies and gentlemen, now I would like to invite His Excellency Professor Dr. Syed Javed Khushi, Consultant to the Coordinator General, YC Comstech and STI Policy Research Center, as well as CPD. Science Communication Diplomacy Journal of YC Comstech. Sir, the floor is yours.
Bismillahirrahmanirrahim. Distinguished speakers, today we have with us Gary Caldwell, Dr. Stephanie, and Dr. Muhammad Ali. Esteemed colleagues, ladies and gentlemen, when Rakib asked me and told me about the topics, I felt it's a very good topic, and it is really my pleasure to open this workshop today on blue technology and health. If you see, the blue technology is the field which is not explored a lot in many countries, especially we have a lot of reserves here in Pakistan. And when you mix the blue technology with health and environment, it gives a lot of great work and impressions. So it is really my pleasure and to say that it is a good thing to highlight this new field in Pakistan and beside Pakistan in the developing countries and in the countries, OIC countries too. It is important to say that we should work on the specific values of this field and its relevance to the contemporary global challenges. Blue biotechnology offers concrete scientific benefits grounded in marine biology, actually, biochemistry, genomics, and everything is there. When I talk about biotechnology or biochemistry, or when I say blue technology, actually I'm considering all these 3-4 fields which are attached to it to get the benefit out of it. Marine organisms provide a rich source of novel bioactive compounds with demonstrated potential for pharmaceuticals, basically. The field of marine biotechnology in case of pharmaceuticals, I will say, is unexplored, including antibiotics. We can work on antibiotics, antiviral, anti-inflammatory agent, and anti-cancer molecules. And my interest and my emphasis, if I will talk about basically the anti-cancer drugs. We can find out many anti-cancer drugs. But here, I have some reservations that we don't reach to those heights to patent our research or to get benefit out of it. I will say both things. Okay, so we have to, in addition to marine microorganisms, there's a lot of organisms also attached to this blue biotechnology. We contribute to advances in industrial enzyme diagnostics and bio-based materials that are more efficient and environmentally sustainable. From an environmental perspective, blue biotechnology supports evidence-based solutions for ecosystem monitoring, bioremediations, and climate adaptations. Molecular tools and marine bio indicators enable us to more accurate assessment of ocean health. While biological diverse deposits offer low-impact methods to address pollutions, habitat degradations, and resource depletion. The scientific benefits also extend to the food and nutrition. We have done some work in Pakistan, basically in the coastal area, on the food and ecosystem and nutrition value and nutrition security. Innovations in agriculture, aquaculture, biotechnology, functional foods, and marine-derived pharmacy provides opportunities to improve protein availability, enhance nutrition values, and reduce pressure on wild marine stocks through sustainable production systems. Importantly, these scientific advances are inherently interdisciplinary. They require collaboration between marine scientists, biotechnologists, environmental experts, health researcher, and data analyst. When supported by effective science communication and internal scientific collaboration, such research can be translated into application that directly benefit societies and economies. This workshop provides a valuable platform to examine current scientific progress, identify research gaps, and explore pathways for collaborative research and capacity building and development, particularly with the development in the OIC member states. With this opening remarks, I invite our distinguished speakers, which are two at this time, to guide us through the technical discussion, and I look forward to be focused and significantly enriching exchange of ideas. Thank you so much.
Thank you, sir, for your insightful and inspiring words. Now I am honored to welcome our distinguished speaker, Dr. Gary Caldwell, Senior Lecturer in Applied Marine Biology at Newcastle University and Chair of the Board of Studies for Biology and Marine Sciences undergraduate program. He holds a first-class BSS honors from the University of Wales, Bangor, and PhD from the Newcastle University with 20 years of academic and research experience. The Dr. Gary research focus on algae biotechnology, including use of micro and macro algae for biofuels, bioproductive products, CO2 capture, and wastewater remediation. In close collaboration with the industry, he also studies chemical ecology and ecotoxicology in aquatic ecosystem. Today, he will deliver a talk on the use of microalgae to remediate wastewater stream and industrial scale. Sir, the floor is yours.
Thank you very much. Can you see my presentation screen okay?
Yes, we can.
Fantastic. Greetings, everyone, from cold, grey northeast of England. And I'm absolutely delighted and honoured to be given the opportunity to speak to you, to present work that we've been doing here using, as was mentioned earlier, sort of green biotechnology, but in a much more industrial scale. So that these cases I'm going to talk to you about aren't going to be, for example, cancer drugs or kind of really finely tuned omics-based technologies. This approach that I'm going to talk to you is much more functional, where we're trying to use effectively algae as a living tool to deal with some of the most pressing environmental challenges that not just the UK, but countries around the world face. And I've chosen today to focus on a challenge that is very pertinent to the UK at the minute, and that is the state of our water environment. We are a highly degraded nation in terms of our aquatic and marine environments, and only really in the last 2 or 3 years has the government and society really woken up to this challenge and decided we really need to make a concerted effort to improve the world that we live in, improve our waters, because environment and health, you cannot separate the two out. They are inextricably linked. So I'm going to talk to you about how we can make algae do this at an industrial scale. And I also want to press home the point that I focused on industrial scale because I want to approach this with financial stability in mind as well. Whenever you're developing products or processes to make a change in the real world, to leave a legacy, it's only really going to happen if it can make money on the same track. So there'll be that sort of economic element also running through the examples I'm going to show you. And you'll notice the second heading, I've kind of strangely named it 2 states, 1 mission. And the 2 states I'm going to talk about will be the 2 different ways that we are using algae to deliver our mission. And they're slightly unconventional in the approach that we take. So, how do we go about this? Particularly as academics or researchers who don't necessarily have, a big financial backing behind us, but we have big ideas. The first thing you want to do is you want to identify what we call the pain point of the industry or sector you're working in. What are the aspects or challenges that these industries or sectors face that effectively keep the big bosses up at night worrying? What are the big challenges that they must meet, but they are struggling to meet with their current know-how or with their current technology? And you go to them with your ideas, but you must, first of all, clearly identify where those pain points are. Otherwise, it just becomes a conversation that never really goes anywhere. So I'm going to talk to you about how we have identified those pain points in terms of the UK water sector, and specifically how we are using what I call unconventional solutions to deliver this. And this sets what we're doing aside from everyone else who's trying to squeeze algal-based solutions into this really quite hectic and chaotic market. Our out-of-the-box approach, doing things differently, is our unique selling point. And I want to show you how you can take these strange ideas, convince the industry that they're worth investing in, and then talk you through that process of developing robust real-world examples, taking these ideas out of the laboratory into the real world. And in our case, I say we did it in a challenging UK context. It's challenging in two ways. One, it's challenging to grow algae in the UK year round because of our climate. We can do it very well for half of the year, but the other half of the year, it becomes almost impossible to do because of our short days, our cold temperatures, and not very much sunshine. But also challenging in terms of in England and Wales, it is an almost entirely privatized water industry. So companies that manage the water for the population, but in a for-profit context. So they have shareholders that they must administer to, but they're also highly regulated. So it's a very strange industry to work in, and it's not perfect by any way, shape, or form. But it's how can we navigate this challenging environment as researchers to get our solutions in place. And then the next thing I will talk about is once you get your industry on side, how then do you overcome the technical challenges of getting your idea off the lab bench to scale it up to industrial scale? And most brilliant ideas, especially in the algae world, fall and fail at this step. You can have the most wonderful product or process that works at laboratory scale, but getting that to a real world scale that again makes money is a real difficult barrier. And then on that, when you're trying to sell it to your partner, when you're trying to make it sustainable long term, you must know your business model. Where is your industry partner getting their income streams from? Is it a product they can sell? Is it efficiencies in their process? You must know that inside out. So it's a real challenge for scientists to effectively partition our mind and force ourselves to think entrepreneurially as well as being a really good, robust, careful researcher. And then the final bit, I'm going to look at what I see as exciting new opportunities that I think will be particularly important for countries with an arid climate and where water is a different challenge that they have to face, but equally it still needs to be managed. So a little bit about UK context. I mentioned earlier, our water systems are highly degraded. Our infrastructure is old and failing, and we are getting discharge of sewage and industrial pollution into our waterways, into our lakes, into our coastal waters, bringing about extremely damaging environmental damage. This photograph I'm showing you down here, this is Loch Ness. So this is the biggest freshwater water body in the United Kingdom. And this is a satellite image taken just this summer. And it is effectively turning into a massive water body that is covered in kind of almost semi-permanent cyanobacterial algal blooms. And it creates enormous ecological damage. It has a lot of economic impacts, and it's bad for the reputation of the country as well. But how do you get hold and tackle these problems where, in this case, algae is part of the problem? Can we flip it around and make algae part of the solution? Now, I mentioned that our approach is unconventional. The photographs I'm showing you here at the top are the conventional ways, the everyday, the normal way of using algae in biotechnology. You can grow algae in outdoors wastewater ponds, or you can use these very expensive photobioreactor systems that you either site outside using light or use indoors. That's quite well established, but it's hard to make the economics stack up, particularly when you're delivering a low value service like environmental cleanup. So kind of these unconventional approaches I'm going to talk to you about is about kind of breaking away from the tried and tested approaches of growing algae, changing the rules, turning things around the other way and moving progressively towards what I call immobilized algae or living algal materials, pretty much taking the need for big infrastructure out of the equation. So I'm going to talk you through 2 case studies. And the first I'm going to talk to you here is one where we still use algae growing in water. We haven't necessarily made the jump to immobilization yet, but it's on the cards. But I'm including this because this is a prime example of whereby if you know your business model of your partner, you can make this into an extremely viable, investable opportunity. So this is a big wastewater treatment plant about 60 miles south of where I am here in Newcastle. And this plant takes sewage waste from a large population centre and also industrial waste from as far away as Scotland that's tankered in. So there's a lot of toxic material goes into this site. Now, specifically, we are looking at the effluent that's generated through the plant's anaerobic digesters. So whenever they take the sludge from all the different sewage works around the region, they're brought into these anaerobic digesters where bacteria break them down, make them relatively safe, produce methane for biogas, which is then used to heat up the local houses. But the liquid waste that is generated from this process has got extraordinarily high levels of ammonium, approaching 3,000 milligrams per litre. Now, that is enough to kill pretty much anything and everything in our natural environment. So it costs the business about £1 million a year just to deal with the ammonium using conventional technology. And that's quite a drag on the business's financial model. So this is where we come in, because they wanted to find a biological, nature-based solution that could deal with the ammonium, but do it at a more affordable rate than they're having to do using conventional treatment. Now, our idea started 6.5 years ago with a very small tube of algae that we isolated from this site. And in that very short space of time, we've gone from effectively 1 or 2 milliliters of algal cells to this area that I've circled here in blue. This is now an internal undercover algal farm that we've built. If you want an idea of scale, if any of you are familiar with Newcastle football team, this area is the same size as the football team's stadium. So it's a very, very big facility. And we are in the process of commissioning that now. So I'm going to talk you very briefly through how we've gone about that process. And in this slide, I've framed our journey in terms of what we call TRLs, or technology readiness levels. Effectively, how advanced your idea, your invention, your process is from getting out of the laboratory into commercial reality where you can start to generate income. So in that 6.5 year period, we have taken it from early TRLs, effectively, we've got a really good idea, we think this works, let's try and get proof of concept. And that took a couple of years to do. But once we started to get the trust of the water company that actually we know what we're doing and we can do good things for you, they started to in turn invest in us, return that faith by giving us access to research funds, which allowed us to then build something more elaborate in our laboratories to reach technology ready levels 4, and we could look to optimize the process of how the algae could consume all that ammonium. And again, this part is another 2 years worth of work, but the data again convinced the company that these are people who deliver what they say they're going to deliver. And they then funded us to take it through the next 2 technology readiness levels, scaling things up, taking things through continuous production, up to the point where we are now, which is this slide, technology readiness level 8, where we're just about to launch this commercially. To give you an idea of how much algae we are producing, so these photographs were taken last year at the start of October. The construction has progressed quite a way beyond this. Each of these tanks contains 40,000 liters of algae. And we have broken the rules here because all these tanks are internally lit. They're not lit by lights from the outside, which is very inefficient. We have lit all these internally. It's part of our design. And will by the end of this year, this will be fully operational, and we will be processing all of the ammonium generated on that site, and as a byproduct, generating what we estimate to be 80 tons of algae biomass on an annual basis, which will make us the biggest algae producer in the UK. But this is only possible where you identify those pain points, where you build that element of trust, where you show your industry partner that you are genuine and that you're committed to addressing their pain point. And if you win their trust, then they will back you by putting the investment in. So this is a sum total of currently of about a £10 million investment, and they have given strong indicators that they intend to extend that investment to a much bigger scale even beyond this. I don't want to give you too much detail about the internal structure because we are currently looking to patent that, so there is a commercial driver behind that too. But the second example I want to talk to you is even more unusual, and in many respects, I'm even more excited by the opportunities that this presents, particularly for countries that are facing different water-related challenges, where water is more of a precious commodity. We've got plenty of water here in the UK. Arguably, we have too much. But in countries like Pakistan, where water is something to be looked after with great care, the approach I'm going to talk to you about now, I think, has got enormous potential. And this is this idea of immobilizing algae into a living material. Perhaps more accurately, if I phrase this as immobilizing them into a living paint, just like you would paint your walls in your home. So if you visualize that kind of how we're trying to go this, and our approach has actually been inspired by nature, by lichens. Now, I'm sure many of you will know that a lichen is what we call a composite organism. That is a combination of algae, bacteria, and fungi all living together in a symbiotic, self-mutual, self-helping relationship. So we looked at lichens because lichens actually cover about 13 or 14% of all the land surface on planet Earth. So they're very successful in what they do. And we asked ourselves, could we turn this success that Mother Nature has provided and transform that into a commercial vision, a living paint that is more than simply a curiosity at lab scale, but something that can do enormous amounts of work in cleaning up our environment. So our approach was to develop latex-based paints that would substitute out the fungal part of the lichen but keeping the algae in there. So in a lichen, the fungus looks after, protects the algae, keeps it from drying out, stops it from being washed away. And we wanted to duplicate that relationship in our living paint. So again, as ever, we start off small and work our way up. So we actually used, as part of our test solutions, little chips of what we cut out from loofah sponge, which is a type of vegetable that we actually got from our PhD student who was Nigerian. They grew that in their village as a waste product. And it appealed to us because of its porous structure and its slightly water-loving, hydrophilic, which meant it was a really interesting potential material to apply our paint on. And our paint is here. This is what you're seeing at the green. This is our latex paint that's got the algae or cyanobacteria. We can use both, embedded and living within that. And as you look through the microscope, you can see you get really good coverage, and all the cells are packed in really tightly together. Now, this has the benefit that in a much, much smaller area, you can get the same amount of biomass, so the cells that are actually doing the work for you, all crashed in together, doing the work in a smaller footprint, which means whatever you build is going to be cheaper to build, it's going to be safer to build, and it's going to be more attractive to investors. So a good way to think about this is effectively an algal city where everyone lives in very, very close proximity. Now, the process of making this is ridiculously simple. There's no magic bullet to it, so I'm going to tell you how we do it. It is just the same as you painting any wall in your home, as long as you use the right paint. So in this case, the scaffold is the loofah we paint on. It can be any material, really. So we apply our paint with our algal cells in green, and in this case, we only use latex-based paints because they work the best. So whenever you paint your surface, just like you do at home, you allow your paint to dry and the liquid evaporates off. The algae then start to get compressed down with the latex particles starting to pack around them. And then once the paint has fully dried, the latex actually forms a complete layer across the algal cells, now physically doing the job of that fungus. And our algae are held in place. They don't get washed away. And latex is important because it allows water to move across the surface, it allows nutrients to move backwards and forwards, and it allows carbon dioxide to move backwards and forwards. And also importantly, as the latex cures, it dries, it goes transparent. So we lose no light coming into the cells. In some cases, actually, some latexes, you can design them a little bit that does shield a little bit of light. So this is very exciting in environments that have got very, very high light levels, like Pakistan, where actually algae might not like having too much sunlight. So you can actually design the latex to become more or less like sunglasses for your algae, and they're safer and more comfortable in their environment. Now, I've also stuck in this strange image down at the bottom. So this is what we call a Malteser tray bake. It's the type of thing you would make with your children on an afternoon to keep them quiet. My story kind of implies that we get this lovely, flat, neat, careful surface. In reality, it looks a little bit more like this, where you have algal cells breaking through the surface of the latex. That is not ideal because they can be consumed by predators or they can get stressed. But there's a way around this, because equally, whenever you're painting your home, you rarely will just put one coat of paint on your wall, because that first coat of paint never looks great. You will allow it to dry, and then you'll go back and you'll paint a second coat of paint. We do that as well in our process. So these images I'm showing you here, the images that I'm pointing to now, this is one of our materials with a single coat of paint. The green is the latex. Every little orangey red dot is an individual algal cell, and the blue strand is the loofah that we painted it on. So you can see from one coat, there are lots of algal cells still exposed to the environment. And that is a risk because you could lose them from the process. By applying a second coat with just the latex, with no algae, you can still see all algae in there, but they are now protected and locked away securely. They will still do their job just as efficiently, but it means that you will no longer lose them from your living material, which means your living material will last for a longer period of time. And the longer you can make it last, the more money you can make. So longevity of these materials is part of the design process. So how do we use these materials in a challenging wastewater environment? I'm going to give you a different example here. In the first example, we talked about dealing with ammonium as a waste product. Here, I'm going to talk about dealing with phosphorus. Now, we chose phosphorus because the government is currently changing the laws in the United Kingdom that makes it much more difficult for water companies to discharge phosphorus into the environment. They get fined very heavily for that. So that is one of their pain points. They know that if they do not address phosphorus pollution, that they as a business will be faced with considerable financial penalties. And we're talking millions of pounds for every potential pollution release. So this is not a trivial amount of money. So that is, for this case, the pain point that you identify for your industrial sector. Now, in this case, we wanted to push our technology even further. One, the latex that we used in the first example contained ingredients from fossil fuels. And we wanted to transition away to make our own latex paints that were 100% biologically sourced. So we designed a paint that was based using starch, and we functionalized it in a certain way. I can't tell you how we did it because we're going through, again, the process of patenting that. But in the end, we made a paint that when it dried, looked like this. So there is a certain amount of opacity to it. It's not as transparent as the other paints, but the algae don't mind that at all. And then we began that process of scaling up our trials from laboratory up into the real world setting. And this is data that I'm showing you here using artificial synthetic effluents, where we're dosing it with phosphate and training, just like you would train your pet, maybe your pet cat or your pet dog. You train your algae to become very efficient in what it does. So we were able to train the algae to become increasingly aggressive and greedy to consume the phosphate from the water. Then the next step was using real effluent with all the other nasties that go alongside that, making it even more challenging. We used the same kind of setup in the laboratory, using continuous flow through, and again, controlled dosing of phosphorus. And you can see how quickly the algae will then remove the phosphorus from it once they are trained. So there's an initial slow lag as the algae kind of figure out, oh, what's this? What am I meant to do? And then they realize, okay, my job here is to eat the phosphorus. And then they do it extremely efficiently. I've included the bottom graph just as kind of an additional point of interest, because these processes will also deal with other pollution. In this case, this is taking out nitrate from the same wastewater, which is an additional pain point for your industry. And the more problematic pollutants that your biological system can deal with, the more attractive then that becomes to the industry to deploy at a real world scale. Okay, so then how do you take this out of the laboratory into the real world? Now, we haven't yet quite gone to the scale of building a football stadium sized structure, but we're on the track for that. So our jump into the real world setting was a little bit more modest. we built a series of photobioreactors here that we packed full with our treated loofah that had been painted with our algae. Again, you can see it's internally lit. In these ones, we've lifted the lights up so you can see them a little bit more clearly. And we designed this so that we can flow different amounts of real effluent through these and then sort of optimize the process and find out what is the best combination for this particular wastewater site. So I'm going to finish off by showing you some of the data. We're still kind of processing our way through this, but this is data that came off that site in early autumn of 2025. And again, you can see that there is an initial slow lag phase as the algae try to understand what they're there to do. Once they start to perform and actually capture phosphorus, the amount of phosphorus reduction coming in to the reactors and then going out and being discharged to the environment can get reduced by approaching 70% or more. And that is considerable. And that would definitely take the amount of phosphorus below the legal limit that the company is allowed to discharge. And incidentally, we also find that the algae also take out a lot of other pollutants. In this case, I'm showing you a lot of the metals that we've captured. And again, in certain circumstances, actually the pain point of your sector might be dealing with things like metal pollution or other organic pollution. And living materials like I've shown you here are an extraordinarily effective way of dealing with these situations. It's a multi-pollution controlling system that is extremely simple to build, very robust, and can last for a long time. And then I'm just going to round off my talk now by kind of flagging up what I see as some very exciting future opportunities, both in a UK context, but also a bit more broadly. So in the UK, so I've already said that we imported our loofah from Nigeria, which is not ideal because there's a carbon footprint associated with that. So ideally, you'd want to have a material that is homegrown, that minimizes the carbon footprint. We have been working with hill farmers growing sheep in the UK and using their fleece, their wool, because there's a very weak market for wool in the UK, and repurposing that to turn it into a nice woolly living material. And these are some of the examples that we have made in the laboratory, and we can modify the wool to make it an extremely effective material at taking these algae paints. But again, this part is still at the laboratory stage, but this year we're looking to scale that up. But I really want to finish off on this example because I think this is of particular interest to the Islamic world, particularly those countries that have to deal with arid climates. And so this photograph I'm showing you here, this isn't my work. This is some work that I've just picked out that was published earlier, but they have taken an idea of immobilizing algae that they isolate from desert crusts or soil crusts and turning them into these living materials. Now, this is an idea I've been talking about for a couple of years, but because we don't really have access to desert environments here in the UK, we've never really been able to test it ourselves. But conceptually, this is a marriage made in heaven because the algae in these desert crusts are naturally evolved to deal with immobilized situations, to deal with high stress environments. So they should make that transition from living in a crust to an engineered paint with barely a heartbeat missed. And if anyone in the room or listening to this is interested in testing this, I think the potential mileage for a technology based upon desert crust algae turned into these paints, I think is potentially huge and transformational in many different biotechnology contexts. So I would encourage people to kind of think around these ideas because I think this is really, really exciting. This is my sort of reverse of a pain point. This keeps me awake at night, but for a very different reason. This keeps me awake at night because it excites me. And the work I've presented here could not be possible without the help from a tremendous team that I've worked with over those 6 years, including partners from industry. So thank you for your attention, and I'm very happy to take your questions.
Thank you, Professor, for your wonderful presentation. Thank you again for your wonderful presentations. If anyone,
My pleasure.
If anyone have a question, you can feel free to ask to him.
Yeah.
Sir, please come.
Thank you so much, Professor Gary, for a wonderful talk about to remediate the waste treatments, particularly in Pakistan and the Islamic countries. We are facing the dry land, arid lands, and we are also lacking the water every year. So my question is that what's the criteria used to select the algal strains and how it is linked to the carbon sequestrations in the dry land and how we can link this to achieve the circular bioeconomy? Thank you so much.
That is a fantastic question because it neatly encompasses where actually much of the research of these materials needs to come in. Not every algae can go into a paint. We've been lucky in that the vast majority of algae we have used have been amenable to being immobilized into a paint, but some haven't. Some are much more sensitive to being immobilized. We've found actually that diatoms don't like being immobilized. Yeah. So we've found that hasn't worked. The examples that I've shown you in that presentation were either a chlorella species that's got a cell wall that's very robust, or cyanobacteria that can deal with quite rapid changes in the environment. So there are ways to design experiments that allow you to screen algae very quickly. You know, when we do our little screening experiments in the laboratory, looking for good candidate experiments or good candidate algae, we will know by the end of 1 week whether an algae is suitable for immobilization. If it has not survived a week in a paint, that is not an algae that you want to work with. The take-home message to you is, if you want to explore these technologies, use algae from your area that are best adapted to the environment that you want to treat. Chlorella for us is a good go-to organism because it is tough, it's resilient. But the example that we used in the first one where we're dealing with the ammonium, that was an algae that we isolated from that wastewater treatment plant. It had adapted to the ammonium conditions and UK conditions. Chlorella can't do that. So it's always worthwhile actually going out to the environment that you want to help and improve, looking for an algae there, because again, nature will probably have given that algae a little bit of an advantage. And if you can then adapt that algae for your environment, your paint or your material, that will then save you years of laborious research. It can save you a lot of time and can really accelerate you getting your materials out into the market, into the world, changing the world for the better. Too many scientists have got amazing ideas, but the vast majority of those amazing ideas die once they try and leave the laboratory. And I have the philosophy of keeping things as simple as possible. The more simple something is, the idea that you have, the more likely it is to succeed. The more complex something you do is, there are just so many more points of failure in there. So that's my second sort of take-home message. Do not try to be too clever. Even if you are a genius, sometimes the genius comes in recognizing do something simply.
Yeah. Thank you. Thank you so much. Thank you.
Thank you, sir. We have another question. So please wait.
Okay, an environment to
and benefits. Can you discuss some of the value-added things, how we can do it? And third, you talked about phosphorus and getting phosphorus
I'm sorry,
out.
I can't. Can you come a little bit closer to the microphone? I'm struggling to hear.
Okay, I'm saying that besides being the environmental benefits getting out of these energies, that can we add, do something value addition so we can get the benefit out of that? And second, you talked about the removal of phosphorus, and after the removal of phosphorus, can we get benefit out of that too? So we can have those value added.
Yeah, absolutely. So your first point, can we look for added value, sort of more or high value products from this? The answer is yes. So let me give you an example that we're currently going through the process of trying to protect. So I also have another business that I have spun out from my research that is producing a cyanobacterial pigment called phycocyanin. So we have a company that is based up in Scotland that is manufacturing this commercially, but it is using sort of quite conventional photobioreactor systems. I have a PhD student who's just about to finish her work and submit her thesis, and she has been taking those cyanobacteria and mobilizing them into these paints and getting it to produce phycocyanin on a much smaller footprint. And we can actually um, milk, for want of a better term, milk the phycocyanin out of the cells without killing and damaging the cells, which means we can have a smaller process that's producing high value pigments that we can then sell to market. In terms of your second point with the phosphorus, yes. So the way we do this is that once the material has absorbed as much phosphorus as it can, we can very literally take it out of the reactors just by hand, and then we can dry it and grind it up, and we can sell it back to the farmers as fertilizer. And it gives you this really good circular economy because it means that the phosphorus is staying on the land. Where it gets leached off, we capture it again in the wastewater treatment plants and we sell it back to the farmers. So it means that they have to use less fossil fuel based fertilizer on their land. And that is an economic driver there as well. It might not be high cost, but there is a societal benefit to it, and there is a definite pull from the farmers to be able to buy sustainable green fertilizer rather than fertilizer they're having to import from overseas.
Thank you.
Thank you, Professor, for your engagement discussion. Now we'd like to request our honorable speaker to the Speaker Professor Mohammad Ali, PhD Associate Professor in the Department of Biotechnology at Quaid-e-Azam University, Islamabad. He is an elected member of the Pakistan Academy of Science and co-chapter head of the Pakistan Biological Safety Association. Dr. Ali leads the Infection Disease and Molecular Pathology Laboratory, managing BSL-2 facility focus on engaging pathologies. His research spans infection disease, molecular pathology, virology, and epidemiology and antimicrobial discovery. He earned his PhD from the University of Rome, Italy, has published over 100 peer-reviewed articles and received the highly cited scientist award at Quaid-e-Azam University 2022 to 2023. Today he will speak on bat associated virus in Pakistan effort for the pandemic preparation. Sir, the floor is yours.
Thank you very much, Dr. Rakib Al Hassan, for this very nice introduction. I'm thankful to the Comstech for the opportunity to speak here, and we'll be discussing our study on Bates viruses and its in pandemic preparedness efforts for the pandemic preparedness. First of all, I would introduce the bats. They are flying mammals, and they belong to the order Chiroptera. They are the only mammals that can have flight, sustained flight, and they are a very highly diverse group of mammals with more than 1,400 species in this order Chiroptera. They can be classified into mega Chiroptera and micro Chiroptera. So they can be major classification is in two suborders. And they can be found in every continent except the Antarctica. And the reasons are they are not there in Antarctica are extreme cold, lack of insects over there, hibernation difficulties, and lack of adaptation. They have wings for flight. They can use echolocation for the navigation, and their lifestyle is nocturnal. That also and because of they are their habitats are rainforests. They can live in caves and also in urban areas. So in our study, we collected samples from all these three different regions to study the bats viruses in Pakistan. They are insectivorous. So their feeding habits. So they are some of the bats are major chunk of the bats are insectivorous. They consume a lot of insects during their lifetime. They can also have fruit diets, and because of this fruit diets, they can play their role in pollination and seed dispersal, and they can also be carnivorous. A few bats are carnivorous as well. Their importance in the ecosystem is pollination. So they perform pollination, and another important aspect they are playing in the ecosystem is seed dispersal. So they can eat fruits and nuts, and they can disperse those seeds as well. And in this way, they are playing role in biodiversity, maintaining biodiversity, and regenerate forests as well. Another very important role is the insect control. So they are literally saving billions of dollars in controlling the insects in the regions where crops are there or where there are fruit farms. They are important reservoir for the virus because of their different characteristics. For example, flight and migratory behavior. They can cross even continents when they fly. And in this way, they can carry virus from one region to another region. And in this way, they play a very important role as a reservoir of the virus. Longevity is another aspect. They have long lifespan as compared to the other mammals, for example, the rodents. And that also contribute to the important virus reservoir. In hibernation, they come together and they roost together in the winters, and in that season or in that time, they can live very closely to each other, and even different species can roost together. And in this way, they can exchange viruses with each other, and that another aspect that they are suitable reservoir. There are virus recombination can also occur in the bats. Bats echolocation is another aspect where high frequency high frequency wavelengths are produced by using high energy, and during this process they can produce droplets and aerosols, and those droplets and aerosols, because of their high frequency, can be disseminated, and that contribute to the viral dispersal. And another important is their immunologic, their antiviral immunity. They have very few interferon genes, and that contribute to the low inflammation in these birds, and that's immune response is another aspect for their function as a suitable reservoir for the viruses. We are here to discuss one of our findings that we detected birds-associated viruses from Pakistan. And this is our first study from Pakistan to report the birds-associated beta coronaviruses. In this study, we collected the bird samples using VTM, viral transport medium. We collected the sample. We used non-invasive method for the collection of the samples. We just spread plastic sheets beneath the roosting area or the bird's living area. And from those plastic sheets, we collected fresh samples in the viral transport medium in very small quantity, like 300 to 400 microliter. And those tubes were taken to the lab, where viral DNA and RNA was extracted. And if it was RNA virus, we synthesized cDNA, and if it was DNA virus, we directly done the PCR amplification. And after PCR amplification, the amplified product was detected on the by gel electrophoresis and was processed for the sequencing and post-sequencing analysis. So in this study, we collected 70 samples, out of which 40 were from one fruit farming area, that is Swat. And in those 40 samples, all the 40 samples were negative for the coronaviruses. However, we collected some more 30 samples from Islamabad. And out of these 30 samples, 8 were positive, and we performed the nucleotide sequencing and post-sequencing analysis for those samples. And we observed that our study viruses are clustering together with those viruses, with the reference viruses from Italy, Finland, and Switzerland and China, and this have clustering together with the that shows that there is some common ancestry of the birds-associated viruses reported from these countries. We also performed the amino acid substitution analysis, and we observed that there are very few amino acid substitutions like that are shown over here and here, and this one and this one. So we were curious to see whether these amino acid substitutions have impact on the structure of the virus. So we performed the structure analysis. This is the reference sequence, and these are the four are the current study sequence or studied sequences, and there is very little or no differences among the in the structure, where there is only slight difference over here in this one. However, the structure showed consistency for the structure. The structure was similar in addition, we studied the hydrophobicity of those molecules of the protein's surface. And except these few changes, there were no change of hydrophobicity. Considerable hydrophobicity was not found over here. In conclusion, this is the first report that for the beta coronaviruses that are circulating among the Pakistani birds birds, and we performed the assessment of genetic diversity. Because the further studies are needed for further studying the genetic diversity and the interspecies potential for the interspecies transmission of the zoonotic viruses. Another study that we performed over here is studying bat-associated influenza virus, and we performed the same molecular method were used for this study as well. In this study, we collected 260 samples. This is from 2024-25. And in this season, this winter season, we are collecting more we have collected more samples as well. Out of those 260 samples, there were samples from different areas: Lahore, Islamabad, Sawabi, Nowshera, Chakdara, Peshawar, and Swat. These are the major bats populated regions, where only Sawabi, this region, were negative for the influenza A virus, while all the rest of these areas were positive. The birds were positive for the influenza A virus. We further performed the nucleotide sequencing of the M gene of the virus. And our studied, the phylogenetics shows our studied samples are clustering together here, and that cluster is closely associated with another cluster that is reporting reference sequences from Egypt, South Africa, Guatemala, Peru, and Brazil. And that suggests that this different cluster suggests that there is uncharacterized lineage of the birds-associated virus that could be circulating in Pakistan. In conclusion, we reported the influenza A virus from the bears in Pakistan, and confirmed that the bears have some diverse influenza virus in Pakistan. We suggest that there should be urgent need for the expanded surveillance of to study their trans-species transmission or possible spread of these influenza viruses to other animals or human beings. There are some limitations of our study as well. This is the first one is sample bias, because this is the bats are the second largest mammal group after the rodents, and this is practically very difficult to collect all the samples and assess it for the viruses, and that is sampling bias. Sample size and diversity. So we have limited access to diverse group of the species, and we collected relatively small size sample, and that doesn't represent the entire bat population. We could not detect it, or we couldn't characterize the H and N subtypes in the case of influenza viruses. and due to the high genetic diversity in these regions. In addition, another limitation is the focused genetic region. We focused on RNA-dependent RNA polymerase gene of the beta coronaviruses and M gene of the influenza. And there is a very focused approach, and that needs more comprehensive approaches needed for the analysis and additional viral gene. Primer design challenge, we designed a number of primers, hundreds of different types of primers to study these viruses. And still there is, because of the very diversity of the RNA virus, or the genetic diversity of the RNA viruses, there is very little success rate for some of the genes to be sequenced in such studies. Another approach we did, we were curious about what types of bats are there, because there are more than 1,400 species of the bats. So we were very curious to develop method for or to study the bats on their genetic makeup or genetic diversity of the bats. So in this case, we used a non-invasive approach. We collected guano samples by using the plastic sheets beneath the bat's living area, and we collected the guano samples, and those guano samples were dried in silica gel and were taken to the lab and we used a specific DNA extraction method for the extraction of the DNA, bat's DNA from the stool of this and we assisted for the PCR amplification and gene sequencing. And the challenge was to how to study bats without disturbing them. In this study, we obtained samples mainly from this northern region in different areas, and in this we performed the DNA extraction, and that DNA was used for the PCR amplification and post-sequencing analysis. Here we observed the two species. We report the two species of the bats. One is Pteropus giganteus, and another one is Pipistrellus javanicus. Phylogenetic analysis shows that the Pipistrellus giganteus is clustering together with the species reported from Bangladesh, and that shows the migratory pattern as well. We can say that all the way from this region, when winter is here, so due to the cold climate over here in the winter, these bats can travel from this region to India and Bangladesh, and that contributes to the that can be the reason why they are having similarity with those with the bats from Bangladesh. Pipistrellus javanicus clusters with the samples from India and China, and that can also predict that the species can migrate from one country to another country. We also studied the amino acid substitutions, and very few one or two amino acid substitutions were reported over here. I think this one is only amino acid, and these two are the amino acid substitutions, but it doesn't have any effect on the structure of the gene of the protein. Limitation of this study is also small size. We could only obtain 11 high quality sequences from 100 samples, and the limited geographic scope as well. Future work is the more extensive assessment of the bat's genome for the studying the bats in this region. In addition to these efforts, we also were engaged in the promoting sustainability and human resource development through the training and conferences about the emerging viruses. And this was our first event that we organized at Pakistan Academy of Sciences, and and the title was Ecosystem Restoration, One Health and Pandemics during 2022. We reported the proceeding of this event in the proceeding of Pakistan Academy of Sciences. The second event was on epidemic and pandemic preparedness, again at Pakistan Academy of Sciences. And we reported the proceedings of this conference in proceeding of Pakistan Academy of Sciences as well. And we published a special issue of the Pakistan Academy of Science, proceedings of Pakistan Academy of Sciences, to publish the main articles or the themes that were presented in this conference. These are some of the pictures and huge participation of the were there. The third event was training on biological safety and risk management at the Department of Biotechnology, Qaid-e-Azam University. This is the fourth event, which was ensuring biosafety, empowering training and risk management at the Badagali campus of University of Peshawar. We traveled from here in the August hot weather. It was very hot weather over here. We traveled to Badagali to have this training, and we published the proceedings in the proceedings of Pakistan Academy of Sciences. The fifth event was in October 2023. That was on pathogen transmission beyond borders and understanding the complexities of cross-species infections at Pakistan Academy of Sciences. This is the pictorial representation, and we published the conference recommendations in proceedings of Pakistan Academy of Sciences. Also, we published a special issue of the conference proceedings. This was another event that we performed and we done at Skardu, Baltistan. Skardu is very important. Skardu in Gilgit-Baltistan is very important because it hosts very large snow area, and that snow or that glacier is melting, and that melting can be a source of some virus or bacteria that could be there in the glacier and can expose to the people and to the animals and crops as well. So we conducted this workshop on emerging viral infection insights from molecular studies at Skardu, and there were huge participation. Here you can see only the males, while the same number of females were also there. And this is another very important activities that we did in Balochistan province. In early 2024, there were some Congo hemorrhagic fever cases in Balochistan, and that Congo hemorrhagic patients were treated in local hospitals, and some of the hospital staff or the doctors were exposed, and also because the patients and those two doctors that were treating them passed away due to that exposure to the virus. And thinking about this, we arranged a workshop at University of Lorelai, Balochistan, on the experimental and ethical consideration of non-human viruses. And we also organized another panel discussion on non-human viruses as a future threat at the Bolan University of Medical Sciences. This is another very huge participation in this conference and in this workshop, and the same amount of female were also there, but due to we couldn't presented those pictures. And in 2024, we organized another conference on mitigating the threat of emerging pathogens and opportunities and challenges at Pakistan Academy of Sciences. And you can see huge participation in this conference as well. And this was the last activity that we performed last year in January at the Department of Biotechnology, where we are constantly focusing on safety in biological laboratory and constantly organizing such events. This is our group. I'm thankful to all these group members for their hard work and for the team up. Our collaborators are from different regions. There must say University in New Zealand, Center of Excellence in Molecular Biology, Wuhan Institute of Virology, and Quaid-e-Azam University. Our colleagues are there. Our funding is from Higher Education Commission and ANSU and Pakistan Academy of Sciences. Thank you.
Muhammad Ali, I have a question for you.
Sure.
I was just lost in biotechnology, but you brought me back to Wuhan type of thing and pet virology and all these things. I was just wondering, what's your idea about what happened in Wuhan? And they have named that as the main thing, carrying the things, because we are not sure whether it is a biovirus or whether it is a biological warfare or some sort, or it is a man-made, or it is just by chance. So have you studied that the virus they have identified and how you compare with that? And then again, what is happening in Bhavalpur? Have you compared that? Why this is happening? And why always we have H5N1 virus in our Bhavalpur areas? All these things, and the as you should going to declare that as a carrier, because after Wuhan we declared that that is not the carrier.
There are many coronaviruses. Before the COVID-19, there were 3 or 4 different coronaviruses were there in the human population affecting us.
I'm talking about emerging infectious diseases.
Yeah.
I'm not talking about the simple infectious disease.
Yeah, any infection that human have, 75% are zoonotic origin. So that explains how important is the zoonotic source of the viruses. So that is why we focus on the zoonotic viruses when we study or when we speak about the emerging viruses. There are cases vice versa. Some of the viruses are transmitting from human to the animals as well, especially the pets are the commercialized animals. So the human are also transmitting diseases to the animals. However, so far the 75% of the studied emerging are the human infections are of zoonotic origin.
But this is a simple answer, and sometimes you say we want to get rid of things and say it is a zoonotic thing. But it is not proved like that.
Yeah, there could be some intermediate species that we don't know about. So that is one of the thing. But there were MERS, Middle East Respiratory Syndrome. There was SARS-CoV-1 that were previously linked to the birds population. So the SARS-CoV-2 is also that was the coronaviruses. There are more than, I don't remember the number, but there is tens of the coronaviruses that are there in the bats that reported in the bats. So there is more
Why in
probable.
Pakistan, when you are working on such many viruses, why in Pakistan we are not working on the early detection system?
Because it costs very high. The cost is very
Lot
high.
of money.
Yeah. And a single reaction can cost thousands or hundreds of thousands. If you want to characterize, for example, metagenomic analysis is not possible in Pakistan for the viruses. For other human samples, it could be possible, but for the viruses, it is not possible because viruses are very tiny. There is very small amount of some of the virus in our samples. So it needs more population, or we would have to grow it.
Now we have close class 3 and class 4 facilities in Pakistan. We can't say that.
Yeah, but it still needs more funding and more human resource for growing those viruses, and then we could pool it together for high throughput sequencing or high throughput analysis.
And I just wonder, you are working in the university, but why when we are not working in the institute which is named for that, National Institute of Health? So why we are not doing that? What is the problem? Basic.
Yeah, national.
Can you mention that?
In my opinion, National Institute of Health is more focused on the human infections. So if there is an unusual virus reported in human, it is reported to the National Institute of Health. For example, if there is a new strain of the influenza that is circulating in the human population, they are studying it.
Mention one thing which is very important. That you work with the zoonotic viruses, and zoonotic viruses, you are working how it is transferred to the human being. Because the ultimate thing you have to find out how it is transferred, and you want to stop that transfer. This is your work.
Yeah, for the time being, we are just studying and characterizing it. And for the transmission, we need more facilities. Thank you.
Thank you, sir. Thank you so much, sir, for your. Thank you, sir, for your inspiring and engagement talk. Now I would like to invite Dr. Abdul Rashid, Consultant Health and Initiative, comstech to deliver his closing remarks. virtual person to query. सबने ईमेल एड्रेस दिया दिया ईमेल एड्रेस के ग्रुप में कुछ होना चाहिए नो क्वेश्चन का मेरे मेमोन नहीं आना है Assalamu alaikum everybody, distinguished physical and also the virtual participants, speakers, and the colleagues from OIC comstech. As we come to the conclusion of the comstech workshop on blue biotechnology for health and environment, I would like to express my sincere appreciation to all participants for their active engagement, insightful contribution like Javed Khurshid Saab, and commitment to advancing their important emerging field, that is blue biotechnology. Over the course of this workshop, we have witnessed a rich exchange of knowledge and experiences that clearly demonstrate the immense potential of marine and aquatic sources that is available in the water in addressing critical challenges related not only to human health but also environmental sustainability and aquatic development. The deliberation highlighted how blue technology can serve as a powerful driver for innovation in pharmaceutical, which is my field, also nutraceutical, this is also my field, diagnostics, and environmental remediation, and last, climate resilience. Our discussions emphasize the importance of conserving marine biodiversity while responsibly harnessing its resources through science-based, ethical, and sustainable approaches. The workshop arranged by comstech and also Quaid-e-Azam University also underscored the growing relevance of blue biotechnology for OIC member and observer states, which are located in 4 continents and having a population of more than 2 billion. Many of which possess vast coastlines, rich marine ecosystems, and untapped bioresources. We are particularly encouraged by the strong consensus on the need of regional cooperation. Either cooperation should be in Africa, in Arab countries, in subcontinent, Russian states, or the Indonesia, Malaysia region. Capacity building, knowledge sharing among OIC states, as I've already mentioned. Strengthening rich research infrastructure, which is very weak in OIC Muslim countries. Promoting technology transfers. Even the European countries, they did not give the vaccine technology transfer to Pakistan and other Muslim countries. Supporting young scientists like this, and including Professor Javed Khurshid, and fostering very, very important that academia, industry collaboration. Basically, I'm a teacher and also as a regulator in Drug Regulatory Authority of Pakistan. There is a weak
linkages between academia, regulators, and industry. This is unfortunate. comstech remains committed to supporting its member states and observer states by facilitating collaborative research programs, training initiative like this. This is training initiative and platform for scientific networking in the domain of blue biotechnology. The recommendations, I request Rakib that he should document some recommendations and action points generated during this workshop will serve as a valuable roadmap for future initiatives aimed at translating research into the practical solution for health improvement and environmental sustainability. मैं थोड़ा सा नेशनल लैंग्वेज में बात करूंगा और फिर मैं इंग्लिश में कन्वर्ट हो जाऊंगा बिकॉज इंटरनेशनल कि जो ज़मीन पे जितने भी मेडिसिनल प्लांट्स हैं जितने ज़मीन पे हैं वही जो है अगर पानी में जो समंदर में या दरियाओं में जो रिसोर्सेज हैं अगर उनको इस्तेमाल करें तो हम शायद ज़्यादा फ़ायदेमंद हो सकें रिगार्डिंग फार्मास्यूटिकल्स रिगार्डिंग वैक्सीन्स और अदर diagnostics. In the last, I would like to extend my gratitude to the resource person Rakib, organizers, the Aisha and other my colleagues in comstech and also in partner institutions, that is Quaid Azam University, and also the most important, the participants who have given the time to the comstech, to Quaid Azam University and 57 Muslim countries population, whose collective action made this workshop a success. Let us carry forward the momentum generated here today and work together to transform the promise of blue biotechnology into tangible benefits for our communities, our environment, and future generations, particularly for 2 billion Muslim populations. Thank you all, and wish you continued success in your scientific endeavors. Thank you very much. Assalam.
Thank you, sir. We have a great academician in our among all of us, Professor Dr. Mustaq Ahmed. He's the chairman of Plant Science, Department of Plant Science at Quaid Azam University. I would like to invite him to deliver his few words. Please, sir, serve the stage.
Thank you so much, Rakibullah sir. Thank you so much, and respected the chair of the Professor Dr. Syed Javed Khurshid Saab, Professor Dr. Abdul Rashid Saab, and all the participants in persons and in hybrid mode. We have a number of the people from OIC country, which is a very important forum for the Islamic world. So this basically the focus was the blue biotechnology. That is the need of the time, because you know that Middle East, Africa, Pakistan have a lot of coastal range. So we have about 73% resources of waters are in a blue biotechnology. That is the food, that's the medicine, that is the energy that we derive from the blue biotechnology. Because you know that America is famous with the ASP, that is the Aquatic Species Program, but the same resources are in the Muslim world, Islamic world. So we think about to make the coordination with Islamic world for the blue biotechnology, either for the energy, either for the food, and either for the health security. So we have a big resource database in the Islamic world. So this topic is a very important topic and the need of the time, as our respected Professor Sayyid Khurshid Javed Saab and Dr. Abdul Rashid Saab and Professor Dr. Ali mentioned. And also we have got a very excited knowledge about the blue biotechnology, about the wastewater treatment by using the microalgae by the Professor Gary from the UK. So this is the important forum. So we work together for the cooperation, coordination for the not only the blue biotechnology, for the bioeconomy, circular economy that is linked to this technology. So I really appreciate Rakibul Hassan to highlight this very important workshop by the Comstech and the OIC and the Qaid-e-Azam University, and we are committed to work together for this technology. Thank you so much for inviting me, and God bless you.
Thank you, sir. I would like to request Professor Gary, we need more session with you. So hopefully I will send an email to you as soon as possible, inshallah. Now I would like to invite some of name from our participants to receive their certificates from our honorable chair, Professor Dr. Saib Javed Khushi sir, as well as our resource persons. So first of all, I would like to invite Afa Noor. Please receive your certificate. Yes, sir. Then Syed Muhammad Mas. Musa White. Sorry. Musa. Oh, I think it's a mistake. Okay. Shanila. Nadim. Give it back.
The coastal zone with respect to the virus.
Nadim. Nadim bhai. Yes. Umair Rahman. Sorry for spelling because I don't know the Pakistani names. Batul. Batul. Yes. Sorry. Umair Rahman. Faiza Habib. Basically, I'm not from Pakistan, so Umar. Faiza Habib. Tariq. Iza Tariq. Cesar. Shehzad Ayub. My money will help you. Mohammed Uzair. Abdul Nasir. Abu Anasi. Okay, he's not here. Maybe he's not here. He's not there. Dr. Yasir Yaqub. Aisha Inam. Mazid. Javiria Mazid.
Thank you.
Oh. Sajid Ali Shah. Sajid Ali Shah.
Yes.
Mix at the biotechnology, plant sciences, biological, biochemistry.
Faisal Ahmed. Faisal Ahmed. He's not there. Yes. Okay. Mohammed Ali. Now I'd like to request to receive Professor Dr. Mohammed Ali for his certificate from our chair.
State of appreciation. Now you have to change it.
I will. I'll change it later.
Change it.
I'd like to request Professor Dr. Mustaq Ahmed to receive his certificate from our chair. Mustaq Ahmed. Thank you, sir.
Thank you, sir. Okay, request make group photo.
We are requesting to all of us, please come. We have a group photo section. Then we will go to our.
Where will be the photo session?
Yeah.
Now we will change.
Yeah.